Hello students, welcome to this session. I, Dr. Rajeshri MUTHA, MBBS DPB from CPS, College of Physician and Surgeon, Anauti Hospital, going to take lecture on general microbiology. Let's study bacterial genetics. So, introduction. Genetics is the study of heredity and variation, seeking to understand the causes of resemblance and differences between parents and their progeny. So, in 1940, principle of genetics applied, and that led to birth of new branch of science, that is molecular biology. The principles of bacterial genetics deals with the study of heredity and gene variations seen in bacteria. All the hereditary characteristics are encoded in DNA, present chromosomal as well as extra chromosomal. The mechanisms of genetic variations, mutation, and the horizontal or the transfer or exchange of genetic material is the transformation, transduction, conjugation, lysogenic variant conversion, and transposition. So let's say mutation. So mutation is random, undirected heritable variations caused by alteration in nucleotide sequence. So types of mutations are spontaneous and induced. So spontaneous mutation occur naturally in any dividing cells without adding any mutation. Induced mutation result of exposure of organism to a mutation. Example, physical agent, ultraviolet radiation, cytosine and thymine are more vulnerable to UV rays. Chemical agents, alkylating agents, 5-bromo-uracil. First type in gene transfer is transformation. So it is transfer of portion of genetic information through the free DNA. So in that Griffith experiment is there stated that the live non-capsulated strains were transformed into capsulated strains due to transfer of capsular genes released from lysis of the killed capsulated strains. So second is a transduction, transfer of portion of DNA by bacteriophage. So there are two types, generalized transduction. Any segment of donor DNA is transformed at the random. Restricted transduction, specific bacteriophage transduces only a particular trait. Example, lambda phage of the E. coli. So in this you can see bacteriophage. So phage DNA enters inside the host bacterium, then destruction of host DNA, synthesis of virus DNA and code proteins, virus capsid synthesis and virus assembly, then transducing particle cell lysis, phages infect another cell, survival of donor DNA. So integration of donor DNA into recipient chromosome or degradation, but so that is unstable gene transfer or unstable gene transfer or the abortive transduction. Lysogenic conversion, bacteriophage, two types of life cycles, virulent, that is lytic cycle, progeny phages inside the host bacterium, ruptures and release. Temperate or non-lytic cycles, that is lysogenic cycle, host bacterium unharmed phage DNA integrated in bacterial chromosome as prophage multiplies synchronously with host DNA. Example, C. bacteria. So phage-coded toxins are diphtheria toxin, cholera toxin, virocytotoxin of E. coli, streptococcus pyrogenic exotoxin, and botulinum toxin C and D. The fourth is the conjugation. So male or donor bacterium, physical contact with female or recipient bacterium leads to transfer of genetic elements through the conjugation tube. So bacterial conjugation and sex pillars or conjugation tube. E. coli, K2, K12 role of plasmid in conjugation. First recognize plasmid sex factor or fertility F factor. So here you can see donor factor. So that is donor DNA, and then recipient DNA is there. So F plus is donor DNA, F minus is recipient DNA. So pilus connects cells, conjugation tube is formed, and F factor is replicated and transferred uh, to the another recipient DNA. The fourth stage is the transposition. Transposons or transposable elements are the bacterial genes that are capable of intracellular transfer between chromosome to chromosome, plasmid to plasmid, chromosome to plasmid, and vice versa. Process of such intracellular transfer is called transposition. Gene transfer by artificial methods. So it is called genetic engineering. It is referring to deliberate modification of an organism's genetic information by directly altering its nucleic acid genome. And the mechanism is known as recombinant DNA technology. The applications of the genetic engineering: production of vaccines, production of antigens used in diagnostic kits, production of proteins used in therapy, transgenic animals, gene therapy. Blotting techniques are Southern blot to detect DNA, Northern blot to detect RNA, Western blot to detect proteins, Eastern blot, it is modification of Western blot, analyze proteins for post-translational modifications using probes that may detect its carbohydrate and phosphorylation. Difference between mutational resistance and transferable resistance. So mutational resistance involves resistance to one drug at a time, transferable multiple drugs. Mutational resistance, degree of resistance usually low, and transferable it is high. Mutational resistance may be overcome by high drug doses, transferable ineffective. Mutational can be prevented by combination of drugs, transferable cannot prevent. Mutational resistance does not spread, transferable spreads to other cells. Mutational resistant mutants are usually metabolically defective, and in transferable they are normal. Mutational resistance virulence may be low, transferable virulence not decreased.
Hope you find this session useful for your understanding and for your exams. Thank you.